we're coming up with several on our own, but if you have any that you would particularly like in a webinar format, then we'll be glad to consider those. Product development from a regulatory perspective is complicated. There are many approaches to it, and before an integrated approach, such as the one that we like to use in our firm, you have the various technical disciplines, clinical, non-clinical, regulatory, CMC, etc., all acting in separate boxes. We found that the integration of those various technical disciplines is very, very important for providing a smooth transition from early data into the FDA environment. So we like to integrate these disciplines. They talk to each other. They work in close concert with each other. And that allows for an approach that truly encourages non-clinical people to talk with CMC people, to talk with regulatory people, to talk with others in the technical disciplines so that the data can be leveraged to the greatest extent for regulatory discussions with the Food and Drug Administration. So harmony can exist. How do you create reviewable reports from early data? We deal with many, many companies that stem from university environments and may still be in university environments or clinic environments in hospitals. How do you take those early data that lead to a promising nascent drug or biologic product into the regulatory environment? Well, you need to consider the primary and secondary pharmacodynamics in your pharmacology studies, animal studies, in vitro studies. You have to then generate some safety pharmacology in a good laboratory practices environment and toxicology studies, primary toxicology studies in a GLP environment as well. You need some ADME. What happens to the product if it's intended for systemic administration or even topical administration? What happens to it? You need to have some data, preferably in an animal model, that indicates what happens to the product. And then development of bioanalytical methods is really critical. What about metabolites? What about the parent compound and how it is distributed in various body fluids in a systemic environment? If you have a topical product, that still may be absorbed systemically. So you need to know what happens, and you need to be able to measure a product and its primary metabolites in blood and body fluids. Well, what are IND-enabling product quality attributes? And by this, we mean the chemistry, manufacturing, controls aspects of a product. What is the structure of the product? What about purity, potency? How much do you need of the material per gram or per milligram or per ml of nascent developmental product to achieve your intended efficacy? That's potency. Do you need good manufacturing processes and material or non-GMP material? Actually, for phase 1 clinical trials in the United States or under a US IND conducted elsewhere, you don't need GMP material. You need high quality material that is well characterized. You don't need GMP material. For phase 2 clinical trial and beyond, you need GMP material. So this is part of your regulatory strategy and your chemistry manufacturing control strategy that must be considered. You need a manufacturing process diagram for the API, that is the drug substance, and the drug product. And this holds for vaccines, this holds for biologic drugs, as well as for small molecule drugs. So for your very first interactions with the FDA, you need to know and you need to be able to codify how the product is manufactured. What in-process controls do you utilize? Testing of pH, testing of purity, testing of concentration. All those need to be considered even for early product development. Well, we've talked about INDs. when is an IND required? And the answer to that is whenever studies in humans are conducted in the US or under FDA control in other regions, phase 1 all the way through phase 3, you typically need an IND. However, if a drug is approved in the US and clinical investigations are not intended for changes enabling the already approved product or advertising, and if you don't change the known risk-benefit profile by testing, you may test the product without an IND in some cases. And some bioavailability and bioequivalence studies do not require INDs, particularly for generic drug development in some cases. But you do need institutional review board approval and an informed consent form to actually execute these clinical trials not under an IND. Well, how to deal with the FDA? Well, one of the many ways to do is to have formal meetings. A type A meeting is a short-term meeting, that is one that is granted in a relatively short amount of time after requesting from the FDA. And that's to resolve a clinical hold where clinical trial or trials is put on hold by the FDA, not allowed to proceed, or a refusal to file. That is, you submit a marketing application, an NDA or a BLA, and the FDA says it's not good enough for us to review. So a type A meeting would typically be held to meet preferably in person and hash out these disputes. Type B meetings are probably the most common. These are for key milestone meetings, pre-IND, before you submit an IND. You can have it with the FDA. Typically these days, written response only or on a telecon, not in person, where you deal with issues that might hold up your IND or might lead to a clinical hold. So you would hold a pre-IND meeting, and we'll get into pre-IND meetings later in this presentation. An end of phase 1 meeting, particularly for oncology products. An end of phase 2, you're about to enter the last phase of clinical trials, and you hold a meeting to discuss the entire program to date and what you intend to do for your final clinical phase so that the FDA will bless your program to date and provide assurance or not that your program is ready for that final phase of clinical trials. A type C meeting is for any other purpose. You've run into a stumbling block in changing your formulation or going from one form of product to another, or changing the route of administration, and you're going to be changing the product. So this would be a general information meeting where the FDA can provide you advice. One takeaway from this entire slide is to remember that meetings with the FDA are intended to help you, the sponsor, not so much the FDA. So you get out of the meeting what you put into it, and you need to plan out a meeting carefully so that you get the maximum benefit of the FDA's advice during the meeting or other consultation. The pre-IND meeting, this is an essential communication with the FDA. And the role of a pre-IND meeting is to identify studies that will support the initiation of clinical trials, which de-risks the IND. And this can also avoid unnecessary non-clinical studies. We see so many programs coming from universities, and very often the university people are geared to generating pharmacology data. Every month generating pharmacology data to write publications, to get the interest of other researchers, and to add knowledge to the field in general. Many of these studies are really unnecessary if you're developing a new drug or biologic product. So you have to carefully titrate your academic needs against the needs to move ahead, get on with it, and develop the product. So you do what's necessary and sufficient and no more to generate pharmacology data. When you're in a developmental paradigm that involves a new drug or new biologic drug. You would also have a pre-IND meeting to resolve potential safety issues. Is the safety margin of this product good enough for use in seriously ill patients? Is this product safe enough to go ahead in non-high-risk disease states where your risk-benefit ratio may need to be different than for a product intended for serious or life-threatening illness. You'd have a pre-IND meeting to discuss available methods to enhance your development, orphan drug, fast track, breakthrough therapy designation, and the animal efficacy rule, which is an availability of a program particularly for chemical, biological, and nuclear warfare agents, where you cannot test the product in humans for efficacy because you would kill the patient in doing so. So you can use human safety data, and you can use animal efficacy studies, carefully construed and carefully designed to show efficacy in an animal model. So, for example, exposure to very high levels of radiation and using an aid that mitigates the long-term safety effect, the short-term safety effects of this radiation. You can test efficacy in animals. And then discussing the difference between a 505(b)(1) or 505(b)(2). 505(b)(1) is an original NDA. A 505(b)(2) is a marketing application where you're actually testing an approved API, an approved active pharmaceutical ingredient for a new indication, a new route of administration, a new dosage form, a new dosage strength, and you can use the data from that previously approved NDA to support safety and other aspects to some extent of your product. So you can actually tailcoat off of someone else's approval. And that's one reason you'd have a pre-IND, to discuss the FDA buy-in that route of administration, that route of submission for your new product application. Typically, this would be done much later in a program, but it could be done to inform your developmental program early on in the program itself. The pre-IND meeting request has these elements in it: the type of meeting objectives, proposed agenda, the list of specific questions arranged by discipline is very important. This is your meeting. You need to get out of it what you want, so you might have questions in all of the areas: CMC, non-clinical, clinical, regulatory strategy, or you might have questions only in some of those disciplines. So you can determine what you want out of the meeting and then arrange the questions to fulfill that need. The quantitative composition of the proposed product, the indication or indications and dosing regimen. Those are the kinds of things you put in a pre-IND meeting request, and a request, when received from the FDA, will on average lead to a meeting scheduled for about 60 days out. In these days of COVID-19, it might take longer, or the meeting might be granted as a written response only, where you submit the letter and a package of information and received a detailed written response document somewhere in 30 to 60 plus days from the FDA that would provide the answers to your questions and any other suggestions the FDA will gratuitously and spontaneously make to help you facilitate your program. The meeting package actually contains the information you have to date, pharmacology information, the background of the developmental concept, the full description of the drug substance and drug product, summaries of available pharmacology and toxicology, not study reports, but summaries, and the brief description of the first IND clinical protocol. We recommend for early pre-INDs, phase 1-ish, that you have a full protocol synopsis. But for phase 2 and beyond, you can include the full protocol because the FDA will have much more to review and provide detailed comments to you based on that full protocol. And again, specific questions to be discussed at the meeting will generally be the ones that were in the request letter, but you could have some additional ones as long as they don't change the overall purpose of the meeting, because the FDA will grant the meeting on the basis of the intended purpose of the meeting as specified in your meeting request letter. The pre-IND meeting will typically be 60 minutes. It can be shorter if the questions are answered and there are no additional comments from either the FDA or the sponsor. We've seen pre-IND meetings last 15 to 20 minutes. But typically they're 60 minutes. If there's a hot topic which the FDA feels really needs to be hashed out, they will actually sometimes extend the meeting. That's more difficult these days because meetings are often stacked very heavily on top of each other in a typical FDA workday. No formal sponsor presentations are recommended. We do not recommend that Herr Professor such and such, who's a great expert on your product or on the field, be allowed to expound for 20 minutes about the wonders of the product and what he or she has found regarding that product, because in fact that takes away from the question and answers which are the meat of the meeting, the questions asked and the FDA responses to those questions. FDA will not comment on data or plans not in the briefing package. It's okay during the meeting to have some tangential questions spontaneously come up as long as they're related to the questions and the information in the package. But speculating on an entirely different subject and trying to get in more topics in the meeting is generally frowned on, and the FDA will typically not comment. They will say things like, well, we have no information upon to base a judgment, or no information provided in the pre-meeting package on which to base a judgment, and so we'll have to defer our answer until later in the development program. Commitments to the FDA are not required during the meeting. Often sponsors who are not used to dealing directly with the FDA don't realize that if the FDA asks for additional studies to be performed, you do not have to commit at the meeting to doing studies. However, if you do commit, that represents essentially an agreement with the FDA. So if the FDA says, well, we'd like to see a carcinogenicity study sooner rather than later, you can say, we understand the issue, we'll get back to you on that. So there's an iterative approach to dealing with the FDA, and it's not cagey, and it's not sneaky, and it's not non-responsive. It simply means that you don't have to commit to something as a sponsor you're not ready to commit to at a pre-IND meeting. Well, the pre-IND meeting is one of the key events leading to a go or no-go decision for some sponsors. If you're very tight in funding and the FDA gives you a laundry list of studies they'd like to see before you can start your program in the clinic, you might have to rethink your product profile. You may have to rethink your indication or indications. You might have to rethink your entire program because you just don't have the money to. Or you may in fact get a greener light than you expected and be able to move ahead more quickly, less expensively, with less time and resource expenditure. So the go decision making should actually be done throughout a program. You should not buy into your concept to the point where you only believe the wonders of your new product, but rather realistically assess what it says and what they show before continuing to make decisions about moving ahead breakneck into the latter phases of your first clinical trial phase or later in the program. So you need to really assess the market. You have to make realistic financial and profitability projections. Make sure your product does not look marginal vis-a-vis the extant data. And again, that's part of the difficult go-no-go decision making. And sometimes you have to say no-go. If you're a leader in a new area, be aware that this is a double-edged sword. The FDA may be extremely interested in your platform, in your product, in your route of pharmacodynamic action, in how your product actually interacts with a particular disease. But if they haven't seen this kind of product before, they may be a bit cautious when it comes to safety support. So maybe need to be prepared for the FDA being very excited, but at the same time issuing some cautions regarding safety support. And if you're a follower, please make sure that you don't ignore known issues with other products, thinking that your product gets a buy because it's, quote, different. At the same time, be aware that a previous approval or a previous movement of a product that's similar to yours with a similar indication into advanced clinical trials may not be a bellwether for your product, because the FDA will consider each product, each structure, each indication product combination as an individual program. So you can't get either lulled into a false sense of security, nor should you be excessively alarmed by FDA concerns regarding previous programs, because your program may have more positives in it. So you need to consider your product individually, just the way the FDA does. Some modern regulatory strategies. Again, realistically review your product indications. Don't ignore emerging safety signals, and at the same time, assess emerging efficacy signals that could actually lead to the change of your program, maybe movement from a general indication to a subset indication where your product shows more promise in a first clinical trial. You have to go where the data go. Let the river flow where it's going to flow, and take advantage of those signals that it provides you. Meeting often and early with regulators is important. Rather than avoiding the FDA, you should be eagerly embracing the FDA, because they really are there to facilitate your program. In the last 20, 30 years, the FDA's mandate for facilitating sponsor programs is even stronger. Congress has endorsed this. The funding that the FDA gets through user fees is intended to facilitate programs and help get those programs on the market cheaper and quicker. And so the FDA does have a mandate, not just to raise cautions and say no, but actually to say yes as often as they can. Consider new clinical trial approaches. Phase 1 expansion cohorts are done all the time, particularly in oncology. Multiphasic protocols where you combine clinical phases, so that instead of going discretely from phase 1A to phase 1B to phase 2A to phase 2B to 3A to phase 3B, this is all becoming amorphosed, to use the term, into preliminary or exploratory and confirmatory clinical trials. And you can have an exploratory phase with a confirmatory phase following directly after the data are initially reviewed in the first phase of your clinic. So this presentation today is intended to deal with early programs, but in reality, early programs often lead to later very quickly. If you see, for example, strong signals of efficacy in a phase 1/2 trial in oncology, you can often go very quickly into a confirmatory phase and perhaps get an accelerated approval. And that's something which I'm sure you're all aware of, but you should take to heart that you don't have these discrete clinical phases anymore to the point where you slavishly have to follow 1A, 1B, 2A, 2B, 3B, 3A, 3B, et cetera. But rather you can combine them in some cases. Adaptive designs, targets and markers across tumor types. This is a very exciting situation that's occurred in the relatively recent times. The FDA is no longer looking at a tumor which comes from a primary in the lung and metastasizes to the liver or the brain as a completely discrete entity from those metastatic tumors, but rather if the tumor markers and targets are on various tumors types or tumors stemming from different organ etiologies, you may be able to actually target a multiplicity of tumor types based on targets and markers that can be measured. And that's personalized medicine leads to that, and also this issue of trials that make sense in the real world, that have real world evidence associated with them, in addition to the formal typing and subtyping of tissues, organs, and tumor etiologies that used to be the hallmark of cancer research. So remember that there's a lot of flexibility. As long as your data support what you want to do, the FDA is likely to be very much in favor of such approaches. Fast track designation is something which can be granted from the moment you submit an IND, depending on particularly animal data, to some extent in vitro data, but primarily animal data if you have no previous human data in your IND. Fast track designation is intended to provide special facilitation of products intended for serious illness, particularly with an unmet medical need, where there's early clinical or preclinical promise based on your data. The request for designation is the formal submission, and there's a 60-day review window for a fast track designation request. The beauty of fast track is that the FDA will meet with you more frequently than they normally do, will take phone calls from you perhaps more easily than they normally would with their busy schedules, and in general provide you a facilitation for your program. Also, when you have fast track designation, once you get closer to the end of your program, the FDA will entertain a rolling marketing application, which would allow you to submit pieces of your marketing application when they are complete, instead of waiting until all the pieces are complete. So the CMC, for example, that's module 3 in the CTD format, which we'll briefly mention later. Module 3 may be submitted in its entirety before anything else. Module 4, the non-clinical, may be submitted before anything else. So it speeds up the FDA's familiarization with your marketing application. Breakthrough therapies is like a super fast track. This involves, again, everything that fast track does and then some. You have to show in your breakthrough therapy designation request that you actually have substantial improvement, clinical improvement, over existing therapies in comparison with any U.S. approved therapies. And the hook on this is that you can show either better efficacy or better safety or both. So you can have equal potency, equal efficacy of your provisional product against approved therapies in the U.S., but if your safety profile is substantially better from a clinical point of view, you can get breakthrough. Most people think of efficacy, but it can be efficacy, safety, or both. But the key here is you have to compare your product against approved therapies. Now that can be approved, that can be licensed in the case of biologicals, but essentially something on the market in the U.S. Typically with breakthrough therapies, the FDA offers a 15 to 30 minute telecon within, give or take, two weeks of you requesting breakthrough therapy designation. And that's a quick telecon that is based on a 2-page summary of your program that the FDA will review, and they will provide advice on this telecon that we suggest that it is your program is ready to be submitted for breakthrough therapy designation, or they will say, we don't think you have enough data yet, you need more patients, we'd like an additional biomarker of support or additional clinical marker. So they'll give you a tip-off as to whether your breakthrough therapy designation full request is going to be successful or not. You don't have to take their advice, but generally it's wise to do so, because otherwise you spend time preparing and submitting a full breakthrough designation request, which might be 30 pages, that really isn't going to do the job for you. So again, breakthrough therapy gives you a dedicated team. Usually a division director or associate division director will be head of this team. Breakthrough gives you a facilitated approach. They'll meet with you and discuss with you ad infinitum your program, help design your final clinical trial if needed, and really work with you. Breakthrough therapies designations lead to a very high probability of success, including accelerated approvals. So if you get a breakthrough therapy designation, it means that your program is very hot and there's a high probability that it will be successful. For clinical trial initiation under an IND, well, this is the IND contents. Now, this is the old regulation which has never been changed. In fact, all commercial INDs, there are two kinds of INDs, primary INDs. One of them is a commercial IND, which a company sponsor will be submitting. Then there is the investigator IND, where an individual investigator wants to treat a small number of his or her patients with an experimental drug product. And the INDs are the same. The FDA review of those INDs is somewhat different. The investigator IND gets a bit of an easier pathway because it's a small number of patients and an individual investigator without the resources of a commercial sponsor. However, they still want to make sure that your safety data and pharmacology data support your program. The investigator IND does not need to be submitted electronically, and so we submitted in this format. The commercial IND must be in electronic format, and it must be eCTD, which is the electronic common technical document, which contains all of these elements as listed here, but in modules, module 1, 2, 3, 4, 5. And this is these modules. This is the worldwide accepted common technical document. Module 1 is the region-specific administrative forms, certifications, the investigator brochure, and the investigational drug labeling. Module 2 is the summaries of each of the technical sections. 3 is quality, 4 is non-clinical, and 5 is clinical. And so the commercial IND must have this format. The investigator IND, it's helpful if you have this format, but you can use the old format, which is still in the IND regulations in 21 CFR 312 at Siegel. So what do you need for manufacturing and controls, product quality? You're a university, you're a small sponsor, you're using consultants to create your product, consulting manufacturers, contract manufacturers, what have you. So a typical phase 1/2 IND package includes documentation of GMP or GMP-like clinical, remembering that you don't need GMP in the US for your first early clinical trial or trials. You need characterization and analytical data, strength and potency information, stability data sufficient to cover dosing. Might be a month, might be 3 months, but you need stability data. The manufacturing process diagrams I mentioned earlier. In-process controls, very brief mention. We check the pH here. We check the weight here. We check the volume here. We do HPLC analysis here. We check for impurities here. Very simply stated. Characterization. Again, you should have a stability indicating analytical method. This is one of the things that stymies very early university or small sponsor trials. Moving ahead is having a stability indicating analytical method. If you don't know your endpoint for stability in terms of analytical measurement, how can you determine the stability? You could be giving to patients something which has fallen apart or metabolized into a completely different structure or broken down into nothing. And then you're giving the patient nothing and you won't see pharmacological activity. So you need stability indicating analytical method. You need identity and purity, assay definition and validation. Again, sometimes you can get away with qualification, not validation in the early stage. So there's some flexibility there, but you need to look at these things, including the impurity profile for the drug substance. Manufacturing changes in the product can result in contamination. So that's important to look at. And accelerated stability through stress, that is high temperature and humidity, are important for storage, for use of experimental product in a very warm environment, particularly. The warmer the environment, likely it is a product will break down. You need to look at issues such as photostability because your product could be broken down by light. So while these things seem like they would only be needed for a product approaching approval, you actually need to make sure that your experimental drug product is at least cursorily tested for these attributes. The formulation for GLP toxicology testing, well, what's the difference between toxicology testing and human early clinical testing? Well, the answer is ideally the test article, your provisional drug product, should be used in your primary toxicology and safety pharmacology studies. So you have no concordance issues between what you use in the clinic for your first, maybe second clinical trial and what you've tested for safety. That way you've got complete concordance. A detailed physicochemical characterization is important. Again, if you don't know what the animals got, then how do you know whether the safety profile in animals is going to reasonably attempt to predict what you generate in your first human clinical trial or trials? So you need to remember these are not just boxes which are checked, but they're very important developmental milestones in looking towards the clinic. You go from the laboratory to testing in a pharmacology model, looking at some PK, ADME, looking at toxicology and safety pharmacology, and then for human clinical testing. So you've got a certain concordance approach throughout your early program. Impurity analysis is important. I mean, obviously, if you store bulk material lyophilized, you're likely to have much less of a degradation or impurity generation than if your product is stored in solution. So you need to consider that in your nascent provisional product development. So the CMC package for the IND itself would involve full ICH stability testing for drug substance and drug product, preferably actual GLP material to be used in the tox studies and the clinical trial material as well. So again, you have that concordance, and any differences are fine, but they need to be put in a table so the FDA sees what your tox material looked like versus what your clinical trial material looks like. And developing the prototype clinical formulation is obviously critical. You can have powder in a capsule for your early clinical trial if it's an oral product. If it's injectable, it's typically a simple injectable formulation. There may be a stabilizer, there may be a pH adjustment, you may be using phosphate buffered saline, but in any case, that prototype clinical formulation needs to be shown to be concordant with what you tested in animals, because then you can have some reasonable predictivity, however poor predictivity sometimes is between animal studies and human clinical data, but you need to make the attempt. Stability deficiencies can cause difficulties in health and shelf life, difficulty in the formulation definition because it's changing all the time. Degradation after dosing is important because you may be introducing toxic metabolites. Again, these should be assessed in as good a model as you have in animals. The choice of animals is important. It has to do to some extent with, again, predictivity in humans. So you need to use animal models which are reasonably predictive of what you expect to see in humans. And to some extent, that's as simple as determining that the metabolite profile in the animal is similar to what you see in human hepatocyte preparations. And that's an in vitro study that you can do to look at metabolism by liver enzymes. And so again, you're looking for as much concordance as you possibly can. Small firms need to minimize costs often and maximize manufacturing data for clinical development. So carefully consider your options. Do you go with a GMP manufacturer first off? Do you need 12 months of stability? I mean, we've seen situations where contract manufacturers love to generate a year's worth of stability before they'll turn over your provisional drug product to you. Well, you don't need that. You might need a month or 3 months more comfortably for your first human clinical trial. So you don't need a year's worth of stability or longer. You should continue your stability for a product where you have a reasonable feel that your formulation is going to be stable or is going to remain the same, shall we say, for the next clinical trial. But you really need to consider how long your manufacturing run needs to be, including your stability, before you can actually use it. And the cost of analytical data being generated periodically, your long-term stability has to be balanced against your need and desire to get into the clinic before your hair turns long. So again, this is all part of the equation in determining your best move forward. Preclinical, non-clinical studies that support early clinical trials. We use the term preclinical, non-clinical carefully. Preclinical is animal data or in vitro data generated prior to the start of clinical trials for your product. Non-clinical includes preclinical, but it includes the generation of data throughout your program. And make no bones about it, your preclinical toxicity studies and safety pharmacology studies, et cetera, are only the beginning. Non-clinical work is typically done throughout a development program to the point where if you need carcinogenicity data, that may be the last data package that you generate before submitting a marketing application. Our general requirements, there are some differences between small molecules and biologic drugs. Vaccines have their own studies, most of them the same, but some a little different. The urgency for serious illness with unmet medical need may result in the waiver of some FDA requirements, and that's very typical. It's also typical in Europe, and it's something you need to consider when you're looking at indications. If you've got an oncology product, dealing with pancreatic adenocarcinoma is a bear. Clinically and in terms of the typical outcomes, it's a very significant fatal disease in the vast majority of cases. And it's a difficult indication to study because efficacy is not readily seen. However, if your product has a particular targeting capability for one or more targets in pancreatic adenocarcinoma, and you see a very strong efficacy signal in your pharmacodynamic studies, you might want to target that indication because, in fact, there is a terrible unmet medical need, and you'll get investigators sitting up and begging, and the FDA as well. So the choice of indication, therefore the choice of your preclinical testing models are important. We mentioned earlier toxicology and safety pharmacology, which are intended as the primary safety studies for support of human clinical trials, must be GLP compliant. If in fact there's a deviation from GLP and you can explain it adequately to the FDA, they will accept the studies, but you should go in assuming that you need full GLP. There are some studies that employ specialized test systems which just cannot, by their technical nature, fully comply with GLP, and those are acceptable to the FDA as long as you identify the areas of noncompliance and evaluate their significance related to the overall safety assessment engendered by the studies. Pharmacology studies do not require GLP, and that's something that I'm going to pound again. Pharmacology, which are the efficacy pharmacodynamic studies, do not require GLP. So someone that tries to sell you GLP for a pharmacology study and charge you 3 times the amount for doing it should be rejected. There are maybe some reasons why you might, particularly for your program, want GLP, but you need it. So what is GLP? Well, just very quickly, it's an entire system of documentation, animal husbandry assurance, very careful monitoring of the animals during testing, reports that have a very complete and thorough raw data section, tables, figures, listings that are exhaustive, and mostly checkoffs for the participants in the study, those who are running the study, double signatures, very careful documentation and representation of quality. Pharmacology studies there are no standard protocols for them. You can do a pharmacology study in your university laboratory. It's a very high quality and really profiles the efficacy of your nascent product. So because of the very, very variable nature of pharmacology and pharmacology studies that are conducted, you really don't need GLP, and there's no way of standardizing them very easily. There are vendors that will do GLP pharmacology. They're very few and far between. General preclinical, remember, the mass of toxicity by corporate euphoria and minimal evidence of biological activity. So you get very excited about something. You say, it's safe. 3 mice didn't die. That's not going to do it for the FDA. The establishment of no observed adverse effect levels, or MATE, which is a minimal biologically effective dose, that would set safety margins for the starting dose for humans. Very important. The FDA will expect that. Substantiation for the clinical doses to be used should be established by tox and safety pharmacology mixed with your pharmacodynamic studies. And you come up with then a safe starting dose for humans. And you need to sell the benefit and risk to the FDA. I get much what we've discussed, and it's just more detail on what the package typically includes in the IND. The toxicity study duration, again, has to do with the proposed duration of the human study and then the suggested duration of toxicity studies. And these are guidances, worldwide guidances under the International Conference on Harmonization, ICH, for dealing with these kinds of studies. These requirements are sometimes waived, certainly for chronic administration of a very highly active oncology drug for long periods of time in human. You often only need 28-day study tox forever, and that's it. Sometimes it's 90-day, but it certainly isn't the 6-month rodent and 9-month non-rodent, which is typically specified here. So there's a great deal of inter-individual waiver that can occur based on the indication and the specific product you have. Safety pharmacology, again, you'll see this on the slides when you get them. These are critical organ systems, single dose studies that examine effects on the activity of these specific systems in animal models. And other preclinical studies may be required based on the product, based on whether you're a small molecule or a biologic drug, and also in case of human tissue cross-reactivity biologics, target a particular cancer cell type, but it also attacks the kidney basement cell membrane. That's very serious. Cross-reactivity is often done to assure that you're not creating havoc in off-target locations in the body. Clinical trials that carry a punch with regulators and invest, very, very critical. You need to make sure that the protocol is concordant with the other information that you've already generated for the program. It can't be a disconnect from your safety pharmacology and pharmacodynamic data. The safety signals, once again, are very, very important. You have to assure that your safety monitoring in your clinical trial targets the potential target organs of toxicity identified in your animal work. You have to make sure that immunotoxicity, which usually is not a big issue with small molecules, but can be for biologics, are addressed. And the data are the critical determinant, not theory, not hypothesis, but actual data that you have generated. There are some special issues for biologic and biotechnological products. This is some general information on what CBER at the FDA regulates. Discussing CMC plans with CBER are really critical. The biologic CMC is much more twitchy than the small molecule CMC, which typically involves synthesized molecules. And so these are some key issues. Impact of changes on minor changes in structure, the impact on safety, very important in biologics. And reproducibility for biologic manufacturing is really critical, and that needs to be documented through controls during the production process. Small sponsors, issues, top-heavy management versus executional internal people, the dynamic tension there, and the importance of using key outsourced providers as team members. Cost versus speed versus resource allocation. That's an equation that's very difficult to assess sometimes and to deal with. Excessive secrecy versus appropriate use of outsiders to help you move your program ahead. Your belief in concept versus hard data. I mentioned that earlier. It's common to small companies. You're true believers and you don't want to give up based on data, or don't want to change the program based on data, but sometimes you have to. And with that, Q&A, please, and feel free to use the chat function on your system, and we're ready for Q&A. A question on the pre-IND meeting for cell and biologic therapies. I didn't include this in the interest of time, and he had perhaps too many slides. The pre-IND meeting, which is called the Interact meeting at CBER, Center for Biologics Evaluation and Research, we've used that very effectively. The pre-IND meeting is typically a targeted consultation that's these days almost always written response, be a telecon, but certainly not in person, whereby one or two targeted questions are provided to the agency. Those questions key for moving ahead with, for example, a manufacturing issue or a clinical issue before actually executing a preclinical study. How can you most effectively design and execute such a study to reach the end that you wish for your stage at that early stage? So we've had quite a few pre-IND consultations with CDER and a small number at CBER since they were established about a year ago. CBER always did pre-INDs, but now they call them interact meetings, and they're more structured. The key for an interact meeting with CBER is probably worth mentioning, and that is that if you basically try and hold a pre-IND meeting under the guise of an interact meeting, the FDA will reject out of hand because they say, no, these are the pre-IND issues. We suggest requesting a pre-IND meeting. So if you're really moved along and you've got quite a body of data, and you tell the FDA that, they likely won't hold a pre-IND or an interact meeting at CBER, but likely they'll push you to a pre-IND meeting. For cell and biologic therapies, the interact meeting is actually quite critical in some cases. If you've got a new CAR T approach, for example, if you've got a gene editing approach, and there's some really gnarly manufacturing concepts that you've come up with early on, an exosome product, then an interact meeting is a good idea, and that would be with CBER. We have a question on patent extension-based FDA classification. I'm not sure what you mean by extension-based FDA classification, but if in an NDA, well, yeah, I mean, there's marketing exclusivity that's 7 years or orphan drug. The marketing exclusivity for a 505(b)(1) NDA, that's first approval of API and first indication, is 5 years typically. And for new indication, previously approved API, it's 3 years. Patent extension is really not what the FDA does. It's really marketing exclusivity. There used to be extension for drugs a long time ago when the Food Drug Cosmetic Act was amended and the 505J official certification came into effect in 1984, Patent Term Extension Act. But typically, you don't really get patent extension. Now, if in fact you're talking about greening a patent and coming up with a manufacturing patent can always be granted. You can get a use patent as long as you fulfill the requirements for a new use of a product, as long as it's not covered by previous patent. And you can green a patent by modifying your product and having the FDA agree with that to the point where you can get basically a new patent. But the FDA right now is not really doing patent extension per se. I don't know if that helps. Questions? Okay. Request for designation. How formal does this have to be? Well, RFD is a request for designation to the Office of Combination Products. It has to do with it has to do with two main groups of topics. One is whether your product is a drug or device, for example, or a biologic or device. Sometimes whether it's a drug or a biologic, but that's been quite well codified recently. The second group of issues that the combination products group deals with at the FDA has to do with a true combination product, which is either a drug biologic, a drug device, or a biologic device. Those are the three types of combination products. And their requested a donation for a combination product as to where it is reviewed, CDRH, CBER, or CDER. You would apply, you usually will come up with your concept that your company would like or your sponsor would like. We believe that this would be reviewed as a device for the following reasons. What is your take on this? And then what will happen is the OCP will actually give you a designation, and that's binding. And they tell you the center, the division, the office, and the contact person who will be your contact person to deal with the regulatory review of your combination product. And it is a formal process. An RFD is a formal process, and it is binding. When the FDA says the primary mode of action of your product is, as you suggested, a device, this is the reviewing division at Center for Devices and Radiological Health, CDRH. This is your contact person, etc. That's binding. If in a discussion at Type C meeting, an FDA official said they would tell, it was English, so I assume that it had to do with this RFD question that I just answered. Stability. Can anecdotal stability of a previous manufactured stocks be helpful? Well, in a very early clinical environment, the information provided for product quality can be of a representative batch. It does not have to be the batch that you are intending to use for the clinical trial. So in a sense, I wouldn't use the term anecdotal stability. I'd rather say if you have stability in previously manufactured stock is here, I would say batch, and you show that analytically your batch, the present batch, is very similar to the previous batch, or shall we say meets the specifications that you have initially established. All drug product and drug substance have to have specifications, and those can be early specifications. Purity 95 to 100%, or 95 to 99%. And if you have a representative batch and you have performed studies and the batch that you intend to use meets the specifications of that previous batch, you may use it for early human clinical trials. That probably answers your question. Well, thank you for your attention. Again, we're not going away, so if you have additional questions, Lauren's contact information is right on this, under this Q&A slide. That would be the best contact, and then she'll get to me. And we're for you. So if anyone would like one-on-one telecons, entertain that very readily, and not going anywhere. We're here to provide whatever we can for the industry.